quick review that we know just about everything about the light-dependent and light-independent reactions that we need. The kind of new thing on Friday, well, I guess it wasn't Friday, maybe it was Thursday we went through this, was the carbon shell game here, the tracking of the carbons, where they have a 5-carbon molecule, perhaps a 1-carbon CO2, and we shuffle that stuff around into a bunch of 3-carbon molecules, use some energy to rearrange them to the point where we can smush 2 of them together and make glucose, and use the rest to regenerate RUBP, which is the CO2 grabber in the first place. That was kind of the last big thing we had, so I want to make sure you're good with that. Any questions? Mm-hmm.
Yeah, phosphate group right there. Where is that?
Here?
Yeah.
It just comes off and floats away and probably gets stuck onto more ADP later. So it's what makes these 12 molecules that have 2 each. 12 of them come off, and now these only have 1 each. Other questions? Okay. Here we go. Cell respiration, chapter 9. It's how we take glucose, which we get from digesting our food all the way down to the simplest sugars. It's how we take that glucose and convert it into energy we can use.
Excuse interruption. Grant Garwood, would you come to the attendance office, please?
In other words, convert it into ATP. Right? The whole system here is geared towards taking glucose and getting as much ATP out of it as we can. How many of you really like balancing equations? Okay, well, I'll tell you there's only 2 that you have to balance in this unit. You've already done 1 of them, right? Here's number 2. See what you can do. The equation for cell respiration unbalanced is this. Glucose plus oxygen, releasing energy, yields carbon dioxide and water. I mean, maybe you're just good at balancing equations. That's how you did this, but most of you, I would think, did this ring a bell? Did this look like something that you've done before? It's like dead opposite, right? It's just the photosynthesis equation, except, right? It's just flipping the reactants and the products, meaning that the reactants of cell respiration are the products of photosynthesis and vice versa. How convenient is that? Like, plants make exactly the stuff that we need to get energy. Extra convenient for plants. Think about a plant who does both photosynthesis and cell respiration. They're making exactly the stuff that they need in a circle. We'll look at that more. Yeah, so it's a relationship. The balance, you knew the balance. It's the 6s again, right? Don't think too much about it, but 6, 6, 6. The oxygen, the CO2, and the waters get 6s. We leave the glucose alone, and it's all balanced. And the relationship to photosynthesis there? Absolutely. They are the exact same reverse. The reactants of one are the products of the other, and vice versa. 3 sets of reactions that we break this down into. We had 2 with photosynthesis. Not quite as neat and tidy as photosynthesis was either. I'm not going to give you the overview diagram until the end because it's not nearly as neat, not nearly as self-contained. The three sets of reactions are named as follows. First, and this is the one that doesn't actually involve the mitochondria, glycolysis. Someone want to tell me what glycolysis means, just based on word roots? You know your word roots well enough yet? What do you think glyco means? Sugar, good. How about lysis?
And the word like glucose.
Breaking down. Lysosome, remember? The thing that breaks stuff down? So glycolysis or glycolysis is just splitting glucose into two pieces. Then you send the pieces, the two pieces that you make, into the mitochondria to do the Krebs cycle, which looks very much like the Calvin cycle or light-independent reactions, but in reverse. And finally, and this is kind of the big moneymaker for eukaryotic cells, it's what the mitochondria is really good at, the electron transport chain, which if you know your electron transport chain from photosynthesis, is going to be a heck of a lot easier. They are almost identical. Today, let's just look at glycolysis. And in this chapter, maybe even more than in last chapter, you need to understand that we are simplifying quite a bit. And so I'm giving you as deep a look as I think you should have without too much clutter. All right, so we're going to keep it fairly simple as far as this glycolysis is going to be another little carbon game like light-independent reactions work. Glycolysis is literally the splitting of glucose into 3 carbon molecules called pyruvic acids. There's the acid part. This has got an O on it, and this is a couple of double bonded oxygens there, making it complicated, but otherwise fairly simple. From C6H12O6. The thing about glycolysis is it requires 2 ATP to even happen, which seems weird, since our whole goal is to get ATP, and here we're spending it. Doesn't seem right. Well, what you need to know beyond that is that you spend 2, but you get 4 back. And so really you're gaining 2. And not only that, you get 2 net ATP, but you also get, and here's where it gets annoying, 2 of a different electron babysitter than NADPH, which is called NADH. If it helps, the way I remember which is that the P is for photosynthesis. So the P is not in there unless it's in photosynthesis. Very similar molecules, very similar functions. They carry excited electrons. This is a process that every cell on Earth does. The next 2 we'll talk about are not. The next 2 anaerobic organisms don't use. In other words, critters that don't use oxygen don't do the Krebs cycle or the electron transport chain, but this is the one we all have in common. Even little bacteria at the bottom of hot springs do this glycolysis. So obviously it doesn't require oxygen. And since all prokaryotic cells don't have membrane-bound organelles, it doesn't require mitochondria either. Happens right out there in the cytoplasm, in front of God and everybody. For the sake of your flashcards, let's do reactants and products. The reactants are glucose, 2 ATPs, and then low energy forms, 2 NAD plus, 4 ADP plus P. Products, we get our 2 pyruvic acids. The high energy molecules, NADH and ATP. And a couple of waters. Again, this is not going to be as clean as photosynthesis was. There's going to be certain products and reactants we focus on and others we kind of ignore. For example, I didn't include the fact that really technically 2 ADP plus Ps are products here, but I don't want to write down all that stuff. Let's just do the carbon game and call it a day. Look carefully, and then I'll have you draw stuff. This one's simpler than the Calvin cycle, so I'm going to have you draw it. Okay, you don't have to draw glucose's structural formula, but here's what it looks like. The glucose, using the energy from those 2 ATP to start with, an investment of 2 ATP, we get a 6-carbon sugar diphosphate, meaning it's a 6-carbon sugar with a phosphate on either end. The phosphate's energy is used to split the thing into 2. We get 2 3-carbon sugar phosphates. Then NAD plus picks up high energy electrons, becomes NADH, and we make our 4 ATP and wind up with 2 pyruvate. Go ahead and draw that. Again, you are not seeing everything here. Obviously, we're just showing 2 3-carbon pieces here at the bottom, when in fact, there's 2 double bonded oxygens and OH and hydrogens. When I draw the real structural formula, we're just simplifying this to a carbon. You can think of this as sort of the reverse of the goals of photosynthesis. Photosynthesis is using the energy from NADH, NADPH, and ATP to pack energy into molecules to make glucose, which is stored energy. Here we're doing the opposite. We're using the energy from splitting glucose to make high energy molecules, to make ATP and NADH. Questions? Ask them if you got it. If you're wondering why on earth we're even bothering with an electron carrier right now, when the goal is ATP, well, the answer is that the electron carriers are going to go up further in our reaction cycle and make a whole bunch more ATPs. We're going to drop those little hyperactive electrons off and get a whole bunch more ATPs out of them than we got here. That's the electron transport. Yeah? It's the energy from those phosphates. Remember we said that when ATP sticks phosphate onto something, that's how it powers a single cellular job. Well, the job here was break that thing in 2, right? So sticking a phosphate on either end provides the energy needed to break those bonds. Don't go any deeper than that. I really can't tell you exactly how those phosphates really do that. That's deep biochemistry that I don't have. You don't need. Any other questions before we leave that? Cool. Let's call that enough for a Monday. I'm not going to add any more work to your load. You have guided reading that maybe you aren't done with. If not, go ahead and use the remaining time to work on that. Keep in mind it's quiz season.